All right. So we're entering near the end of our systems. We've talked about digestive, respiratory, lymphatic all recently. Now we're going to talk about my favorite system, the urinary system. It is super cool. It's some amazing structures, some amazing physiology, and at this point we're going to talk about it in person. But hopefully this is still a good enough experience for everybody. So let's get at it. The naming for these lectures is going to be inspired by one of my favorite movies of all time. Well, duologies, I suppose. Kill Bill Volume 1, Kill Bill Volume 2. So that's where it comes from if you're not up to date on your Quentin Tarantino. So, urinary system. What's the big deal? You make some pee, you move on. Who cares? Well, turns out the urinary system has a very important organ called the kidney. How about the kidney before? But the kidney does a whole lot of stuff for you all the time. And it is a very important organ, a very important structure, and the process of releasing waste is very important. And this is something we do every day, no matter who you are. Everybody pees, right? But what goes into it? Because there's a whole lot that goes into it, and then you can analyze pee as you did in the lab last week, hopefully, in some capacity. You understand like the benefits of urinalysis at least, and then you can understand that okay, I'm starting to put some things together here. This is actually pretty important. And I think the process of how this works is just fascinating in general. And that's a nice bathroom. I think we can all agree. That's a nice bathroom. I've always wanted a big, nice master suite bathroom escape like this. So I really don't care about the guests. You know, they can do whatever they want to. It's my house. I want my bathroom to be, I don't know, some sort of just, I don't know, whatever this represents, something nice, a paradise. I like the tub thing. That looks cool. I like the shower. It looks pretty neat. All the exotic counterware, all that sort of stuff. I don't know. It just looks nice. A good bathroom is definitely a welcome thing. I don't know. Just cool. So, an oasis. That's it. An oasis. I always watch a lot of HGTV and, well, not really that much, I'll be honest with you, but some House Hunters from time to time. And I always enjoy seeing some of the good bathrooms they showcase. And a bedroom and kitchen's important too. A nice living room. But I mean, bathroom's underrated. Anyway, the kidneys. So the kidneys are a big time organ. And they help maintain the internal environment of the body. But how do they do that? Right? Tons of functions. So the kidneys I think of are the Avengers, right? And then the Avengers are this big overall unit, right? They're a major team, and they help maintain the Earth's environment. And they do so by doing a variety of different functions, and those functions can be carried out by, well, in this case, specialized heroes, but sometimes in the case of the kidneys, specialized structures, or just the overall physiology of it in general, right? So kind of the same deal. So one of the things they do is regulate water volume and solute concentration in water. So by getting rid of water, you're getting rid of solutes, or by getting rid of water, you're affecting the solute concentration because you're keeping that volume the same while decreasing the amount of water, so therefore concentration goes up. You can also regulate ion concentrations, and it goes the same deal with water volume, either by getting rid of the ions directly or by affecting the water in the space. And then also the acid-base balance to the whole operation. You got to keep those things in check. So with that system, so if your blood deviates too much, deadly. So this helps with that. You also got getting rid of waste, toxins, and byproducts of drugs you can take for various purposes. So any sort of like byproducts of those things will get released. And then one of the more important functions is produces erythropoietin, which is of course related to blood pressure and making of red blood cells. So we kind of need those things. I think I had those reversed. Yeah. So erythropoietin does red blood cell production, and renin does blood pressure. So flip those around. I'm going to flip those around the notes because, yeah, that's wrong. But I don't know how that happened. That's funny. Well, anyway, that's there. And something else they can do is vitamin D. They can help regulate that whole thing. And you know what? I think I can actually change the erythro and renin, so you may not even notice. So if you're listening to this, and erythropoietin regulates red blood cell production, renin does blood pressure. If it says otherwise, well, what I just told you is right, and what your notes will say is right. Anyway, they also do with activating vitamin D. It's important, and also carrying out the creation of foam. Okay, so the urinary system. Kidneys, a big part of it. Are there other structures involved? Of course there are. Right? You can't just have, one organ being the whole show. You need more than that. And so you also have the ureters, the urinary bladder, and the urethra. All do various processes. So the urethra is involved with getting urine out of the body. Ureters transport urine to the bladder, and the bladder just stores it until it's time for the pee. All right, and so here's a nice diagram showing those structures inside of a body. Here's one on a real body, a little less glamorous, a little gross. So the kidney, if you remember, trying to find the kidney was always kind of tough because you'd look at it and go, oh, I don't see it, but it's because it's retroperitoneal, right? It's behind that layer, so you have to dig through it. The right kidney is a little lower because of the liver being present, while the left is higher. On top of each kidney is a little hat, right? The adrenal gland. We talked about that. We talked about the endocrine system way back in January. Feels like forever ago. And then there's the hilum, which leads to the more medial structure. So it's the one that leads more towards the middle of the body. And that's where the actual blood vessels and the ureters will originate from. So you'll have artery, vein, and ureter at that spot. And so they're going to enter and or exit depending on what the job there is in that one location. And so here's the kidney location. So you can see how high up they go. And so if you have a kidney stone attack, you may feel some pain, and it'll be up near mid-back, and you won't necessarily realize what it is. And then that'll be like, oh, it's the kidney. I'm dying. Or I feel like I'm going to have some intense pain. You can also, if you've ever seen this in boxing matches, they'll go with like those shots right there. It's like kidney punches. So that'll hurt. But yeah, pop them right there in the back. So that's anyway. The kidney does have some supportive tissue to it. And so it has 3 layers of supportive tissue. It's got some cushiony fatty layers, so it doesn't get, damaged, hold it in place, those things. Then it also has a fibrous capsule. And so this helps try and limit infections from spreading to the kidney. So just trying to, protect it. That's the main things these are. So just supportive things around it, protect it, insulate it. That's and so here is a case of the body where you can actually see the kidneys kind of where they're at in relation to the vertebrae, underneath and behind the peritoneal organs. So just cool. All right. So and now we're going to look at the actual internal kidney anatomy real quick. And so we have, like we have with a lot of organs, you have a cortex and a medulla, right? So the cortex is the outermost layer. The medulla is the inner part layer. So it's deeper, using our anatomy terms. Within the medulla, you have these cone-looking structures called renal pyramids. And so that's the little pyramid things. I mean, the picture's labeled, so you can see the regions. Cortex is the outermost layer. The inner part is the actual medulla. And so the broad base covers the cortex while it tips into the area that will end up leading to the renal pelvis. So as it moves towards on the internal side of the kidney. And so that's the tip of the pyramid, and they're separated by inner layers of cortical tissue. Renal columns will dip in there and try and separate them out. So it's kind of, overlapping a little bit, but that's kind of the structure we're looking at. And the bottom left is a picture of the pyramid in Memphis. So, I mentioned that a few times before. There it is. It's been a long time since I was there, but I've been there before. I'll probably never go back there again. The other structure is the renal pelvis, and so this is the part that's near the ureters and that entire area. So it's a funnel, and it's continuous with the ureter. So at this point, a lot of the pee has been drained into this area. It's going to be drained out of the body. The renal pelvis is divided into minor and major calyces. And so these calyces are areas that collect urine. The minor ones or smaller ones, they connect it directly from the renal pyramids, the medullary pyramids. Those papillae will drain right into here. And then these minor calyces will dump together and form major calyces, which will then empty into the renal pelvis in that larger area. So if you think about it in terms of like rivers, so where you have a lot of rivers and these little drainages, and they just empty into bigger rivers, like shown here in the picture to the right. that's kind of what's going on here, is you have, all those major calyces dumping into the renal pelvis, which in this case would be the large, thick blue Mississippi River, and then that will dump out of the body, which in this case will be into the Gulf of Mexico. So that's our ureter at that point. So the urine flow is you go renal pyramid to the minor calyx, to the major calyx, to the pelvis, to the ureter. And you should be able to describe the flow of urine through the kidney. And, well, I guess throughout the body, too. You should be able to do that. So all the steps involved. So make sure you can do that. And also where urine's formed, right? Which structure. Not just the kidney. It could be specific on that. All right, so here's a picture of the kidneys. Hopefully you saw this in lab, so you're familiar with these structures already. But yeah, you can see the columns that they're present, the pyramids, the cortex. The pyramids look much like pyramids, but rather like pink splotches, but it is what it is. Okay, so now for the kidney blood and nerve supply. So it receives blood via the arteries that you see here and the picture at the bottom right. And so blood comes in, it gets filtered out, and it receives a ton of blood. So renal arteries, you can see, do about a fourth of cardiac output to kidneys each minute, which means the kidneys are processing a lot of blood and filtering it out, and then going to absorb things to put it back into blood. So it's a constantly working system to make sure your blood isn't, the concentrations of various substances is okay. Get rid of waste, etc. The nerve supply comes to the sympathetic nervous system via the renal plexus. So just the sympathetic nervous system is one of the key parts to that. So here we see the picture of the kidney with the blood supply to it, and you can see tons of blood vessels in it, the various vine-like structures near the edges. That's where the actual filtering structure is going to be, the glomerulus. And so it's very concentrated there, and then once it gets filtered and reabsorbed, it's drained out via the veins and will leave the kidney and go back to the heart. This is a complicated pathway you don't have to know. It just shows you some of the structures involved and what arteries are involved. I just think it's kind of neat to see like the pathway and how many arteries and sub-branchings there are in the kidney, and it shows you how much blood supply this thing gets. And there's a lot of vessels happening, and it's pretty complicated, the overall flow of the system. All right, so you can't talk about the kidney without talking about the nephron. The nephron is one of the more important structures in the urinary system. So it is a super, super important structure. And so the nephron is the main functional unit that helps form urine and helps the kidneys do its main job, which is filtering blood. There's over 1 million of these per kidney, and there's so many of them, and there's different types of them, there's different parts of them. We're going to be very familiar with the nephron. So we're going to go through all the parts of these things, look at how they work. So the nephrons are we're spending most of our time at in this chapter. So there's two main parts of a nephron. You have the renal corpuscle and the renal tubule. So we're going to look at all the structures. So we have first off, the corpuscle. The first part of the corpuscle is this fun little word called the glomerulus. So the glomerulus is the part where you actually have capillaries coming through into the system. So that's the capillaries. And so you can see them labeled in this picture in the bottom right. And so within these capillaries, their structure has changed a bit. And so they're very porous, which means a lot of filtrate can be absorbed and passed through, which I mean is the intention of capillaries anyway. But especially so here, to really suck out all the filtrate, so all the pretty much non-red blood cells and larger proteins out into this overall structure, into the nephron. So it can be filtered and analyzed to see what needs to be kept and what doesn't. So the filtrate is just the fluid that comes from blood. So that's what that is. So you're getting that taken out. So you have blood coming through, the filtrate is taken out, and that's going to be used and processed to form urine. And that all happens. The other part of the corpuscle is the glomerular capsule. It's called the Bowman's capsule, and that's the actual cup-like structure holding the glomerulus. So it is hollow, and there's two layers to it. I don't think we really get into those structures too much, but in short, they just are involved with the process. So one of the layers helps clean to the capillaries, and the other one helps with filtering blood. So that's what their tasks are. So two parts to the renal corpuscle, glomerulus. And so here's a larger diagram that actually shows some of those structures. And so you can see the part we just talked about, the corpuscle, and you can see the long tubules, which we're going to be talking about pretty soon. I also like this figure because it shows you how the epithelium changes along the way. So you can see just how that shifts as you hit these different sections. And that hints. Okay, so now let's talk about the renal tubule. The renal tubule is where the magic really happens. And this is where I like this thing a whole lot. The tubules in general, on average, about one inch long, so they're pretty short. But at the same time, though, that's pretty extensive for a very small structure, and there's millions of these inside the kidney, so it's pretty remarkable just how small they can truly be. But the tubule itself has a single layer of epithelial cells, so single layer of epithelial cells means great for absorption and taking things in, because that's its job. But each one of them will vary a little bit in the type of tissue that they have present, or like what modifications or adaptations each of those tissues have. There's three major parts, the first of which is the proximal convoluted tubule, also known as the PCT. And it's called proximal because it's the one that's closest to the renal corpuscle, so closest to the glomerulus. Then you have the nephron loop, which is the actual dip where you go down from the proximal convoluted tubule and then up to the other convoluted tubule. And that's where some really interesting things happen in that nephron loop. Turns out that structure of going down then up is super important to the functioning of this thing. And then we have the distal convoluted tubule, and it's called this because it's the one that's farthest in the renal corpuscle. So you go down and up, so you can see it labeled in the picture to the right. So distal refers to like vessel distance, not like actual geographic distance in this case. And then it'll eventually dump into the collecting duct. So that will drain to the collecting duct. It's a different structure though. All right, so the tubule is these three parts: PCT, nephron loop, DCT. The collecting duct is an overall larger drainage system. It's a sewer system that collects everything from many different apartments or many different units or houses or wherever you live. But each house has its own like toilet and pump system. So that's what the renal tubule is. Collecting duct is just a larger structure that collects it from many different. All right, so let's talk about these different structures in depth. So the PCT, the proximal convoluted tubule. And so here they have these cuboidal cells with microvilli that form a brush border. We've seen this before with the small intestines, and it's similar functioning here as it was there, and that the main function is to increase surface area. And so, yay, increasing that surface area, and by doing that, the benefit is it allows you to have more absorption take place. And what do you know? Tons of absorption takes place in this joker. So, yeah, it does so. So a lot of reabsorption and secretion, getting rid of stuff too, happens here as well. And this is only found in the cortex, so that's an important thing to note. Not all these structures are only limited to the cortex, but this one is. And it makes sense because this one's closest to the glomerulus, which is only in the cortex as well. So that helps you see some differences between those. There's the nephron loop. So the nephron loop. The nephron loop is this U-shaped structure, and there's two parts to it, the descending limb and the ascending limb. Each one's different. The descending limb is the one that will go down from the proximal part. All right, so the DCT is the other structure of the renal tubule, and so here you have some cuboidal cells with some microvilli, but not as many, right? So again, shows you more of what's happening. While the PCT is more involved with reabsorption and secretion, this one's really just secretion and not as much reabsorption. The reabsorption can depend on if some hormones are present. So we'll look at this structure being targeted later on. And this is also only found in the cortex. The nephron loop can be found in the cortex or the medulla. It depends on what type of nephron you're looking at. And so here's an example of the microscopy of this thing, and so you can get an actual feel for it. And so, depending on who you may have, so you may have seen this in lab, you may not have, but overall you can tell them apart because they have the microvilli and the PCT and the DCT does not. You can also see the glomerulus. So, the other structure that you have to have is the collecting duct. And so this is the thing that will collect things from the individual nephrons. They receive the filtrate that all those nephrons are forming, and they dump into this larger drainage system. And they have cells that are also responsible for maintaining the balance of various items like water and salts. And so these will go completely through the medullary pyramids, and these are responsible for the striped appearance of medullary pyramids. And eventually these collecting ducts fuse together to eventually form the minor calyces as they dump the filtrate into those, so eventually, well, will be urine. So that's their job. And we're going to learn a lot more about the functioning of these things and how they work. But there's overall structures again. So you see the corpuscle, the tubule, the different sections, and yeah, you see some of the different cellular changes too that happens in them. So these are important structures. You should be able to describe all the layers of the nephron, eventually what they do, and they describe like locations, the pathway of urine as it's being formed. So I'll be able to describe all that stuff. It's definitely going to be on the final for sure. This is my favorite system, right? So there's going to be some questions about this soaker, including the higher level functioning once we get to that. So, to recap, things you should be able to do is you should be able to talk about the structure of a nephron, identify all the uniqueness to it, do this reliably, understand like how these structures work, what would happen if some of them are missing per se, for example, and then also describe the fluid flow through the nephron. How does it go? What structures does it hit? Where are these structures found? All that sort of stuff. So when you have nephrons, there's two types you can have. You can have a cortical nephron or a juxtamedullary nephron. These names are based on where they're found. Cortical nephrons, 99%, most of them are in the cortex. So generally cortex. Very little, if any, part in the medulla. And this is most of our nephrons in humans. How many of these types of nephrons you have will depend on the organism. So if you're a little more heart-strapped for water, you make more concentrated urine, you'll have more juxtamedullary. Us, we're kind of doing okay, so most of ours are cortical nephrons. The other type, though, is the juxtamedullary, and it's called this because it juxtaposes into the medulla. So that's why it has this name. Cortical looks like cortex because that's what it means. And so here you have very long nephron loops in the juxtamedullary, and it'll go deep into the medulla. The importance for this is it helps make more concentrated urine. So the ratio of these that you have will help determine, you can actually study and learn the abilities of the organisms of that organism. So like what's its environment like? Because if you have more of one than the other, it lets you know, it's like, okay, it's a food, or it's a water-poor environment, it's a water-rich environment. So it just depends. We'll look at a pretty awesome example of an organism later on that has some pretty amazing nephrons, including camels. So, yeah. You can see the comparison in the picture. Here's another picture that really drives home how these are different. And so in this one, the cortical actually would go into the medulla a little bit, but not much. Meanwhile, the other one has a extensive dippage into the medulla. Also here, you're seeing the first case of the extra surroundings of the nephron. And so those are blood vessels, and so the nephrons are constantly secreting and reabsorbing things, and well, blood plays a role in that. So things that are reabsorbed goes right back into the bloodstream. So, cool. And that's our next point. So the capillary beds that surround the nephrons. And so not only does the glomerulus exist, there's also other things. So the renal tubules are associated with that glomerulus, but also the peritubular capillaries. And then the second thing they can be associated with is the vasa recta. It just depends. So the vasa recta is for the juxtamedullary, while the peritubular is more for the cortical nephrons. So capillary exchange is important here. And they absorb the things that are reabsorbed by the nephrons, and they put them back into the bloodstream. So now we'll look at the glomerulus and see what makes it so special, and so what adaptations it has to aid in its function. And so, overall, the function of the glomerulus is for filtering of blood. So these capillaries in it, the capillaries that make this structure up are specialized for this. And so they are different from other capillary beds because of this. So they are fed and drained by an arteriole, which is different. Some are drained by, because if you think about a normal capillary bed, it's an artery to vein. This is not that way at all. So the first arteriole we have is the afferent arteriole. It enters the glomerulus and will leave the glomerulus via the efferent arteriole. And that efferent arteriole is either going to lend into become either the peritubular capillaries for a cortical nephron, or to become a vasa recta for the juxtamedullary nephron. And the blood pressure is high in the glomerulus. And normally, again, that's not something you typically see that much in capillaries, but you see it here because afferent arterioles are larger in diameter than the afferent arterioles. So you're changing the volume of the space, which creates a higher pressure. And the arterioles in general have a little higher resistance. So you have this high blood pressure, and that helps drive the pressure of the fluid out. So remember we looked at that equation way back when of calculating that whole thing out. And that comes up into play here. So it drives out the fluid. All right, so peritubular capillaries are low pressure in comparison. They are very porous as well, so they're adapted for absorbing things. So in this case, it's a little less about filtration and more about reabsorption. And so they absorb some things that are taken back in via the nephron, and they come from efferent arterioles. So once that glomerulus pumps blood out, it then meets back around and reabsorbs most of it back up. So it's kind of an odd system. It seems kind of wasteful and a waste of time, but it's how this whole thing works. To maximize and not get rid of a bunch of excess stuff you don't need. Want to recycle as much as you can. So that's what the body's trying to do. And so these stay real close to the renal tubules as they collect things that are being reabsorbed in that area. And then where they empty into venules, which become veins, and we'll go back to the heart. The vasa recta is a different type of structure. And so it is long. It's a long structure. It hangs parallel to the nephron loop of the juxtamedullary. It also arises from efferent arterioles, and they serve juxtamedullary nephrons. And so there's a different type of structure that serves these instead of peritubular capillaries. And they function, and they help function in the formation of concentrated urine. So vasa recta, it's a cool name. And so you'll want to talk about the physiology, you'll understand how that helps in the formation of more concentrated urine. It's all involved with absorbing things and setting out concentration gradients in the nearby interstitial fluid. So that's what that's important for. We'll elaborate on it later. But here you can actually see the craziness that is a glomerulus with the afferent and efferent arterioles. Very zoomed in picture of one. That's kind of creepy. And then again, this picture again, you should be pretty familiar with these. Spot all the structures, functions, etc. And then this one shows you a nice zoomed in glomerulus. And so you can see kind of what makes it different. So it's got some various processes and extensions coming in for filtering. So it's kind of unnerving, kind of creepy, but again, capillary, high pressure, shove that fluid out, keep the blood in. You don't want to lose blood here. If you have blood in the urine, you got something else going on. So, nephron types, you should know them, know where they're found, know their differences. Capillary bed types, right? Vasa recta versus the peritubular. What makes them different? Which nephrons do they serve? And then overall, the structure of the capillary beds. What makes them a little different? So, kidney physiology. A little bit. We're going to begin that, just a hair here. So, a lot of fluid is processed by the kidneys a day. You can see that's a lot of fluid. But despite all that, you only form about 1.5 liters of urine a day, which isn't that much. It would depend a bit, definitely on lifestyle, and like how much water you're drinking or not drinking, etc. But that's just, on average. And so you're filtering all this, blood volume, and your kidneys actually filter the entire blood volume of your body 60 times a day, which is insane. They're doing so much filtering. They don't get a day off either, which is what's remarkable. They're just constantly working. No weekends off for them. Take the weekend off, you die. So, yeah. But the kidneys absorb a ton of oxygen at rest, and that's how you know this is how important this thing is. Almost 1/5 to 1/4 of your oxygen at rest is going to the kidneys. One organ. And the same with the biggest organ. So that shows you how important this thing is. And then the filtrate that gets formed is pretty much blood without the cells and the proteins. So just plasma. That's it. Without proteins, so you want to keep those things in there because they're too big to cross across the capillary membrane. And then urine is formed from the filtrate. And so urine itself is a very small percentage of the original filtrate that comes in. So you can't say urine is made from filtrate, but not quite the same thing because all the filtrate that's made isn't lost. So only a small percentage of it is. And then of that urine, it contains just various waste and things you don't need anymore. So like if you take extra vitamins sometimes, if you see like the daily allotment of like vitamin C or something, like the rest of it can't be absorbed, so it just gets peed out. So like this is the sort of thing it's talking about. So when you're trying to make urine formation, there's some processes involved, of course, and making sure things, go smoothly. The first of these processes is glomerular filtration. And so the idea is you want to try and prevent large proteins and cells from getting inside the filtrate itself. Then the other things you need for urine formation to occur is tubular reabsorption. So you need that nephron to return most of the stuff that's in the filtrate back to the blood. So you would think, why filter all this stuff if you're going to return 99% of it back? But while most of it goes back, it does remove the excess stuff. The only way to be thorough in that removal is by filtering all that filtrate. So just how it goes. And then, of course, tubular secretion. And secretion allows you to selectively move items from the blood to the filtrate. And so it allows you to get stuff in blood out extra. And just maybe there's some extra stuff that's in there. It's like it didn't all get out in the glomerular filtration step. So it's a few checks and balances and fail-safes in the system. And so here you can see the processes. And so again, to form urine, you need to have glomerular filtration, tubular reabsorption, tubular secretion. You can see where all those can happen here. And so glomerular filtration. And so this is a passive process. As we look more in depth into it, no energy is required for this. And so it just does its job. So luckily the hydrostatic pressure, remember that pressure that the blood is exerting on the walls of the capillaries, is high enough that it just pushes through the filtration membrane and pushes that filtrate into the nephron. And so that's good. And then this pressure is high enough that it does not allow for any reabsorption of any of these filtrates in the glomerulus itself. So even though capillaries normally can reabsorb along that pathway, when we looked at that, it doesn't happen here. Pressure's too high, it's moving too quick, so it doesn't happen. It has to be reabsorbed elsewhere. So this is not, the same capillary beds you're used to. It's a little different. So now, at the site of the glomerulus, you have a filtration membrane, and this allows things to pass through. However, it only allows things smaller than blood cells and plasma proteins to pass through. So normally no cells can pass unless you've got an injury or something else like that happening along. Same deal with these plasma proteins. So you can measure and see if those things are present to get an idea of what's going on inside the kidneys. To see if there's an issue of some sort. There are 3 layers in this membrane. We're not going to go too much into some of them, but their main jobs are they're modified to allow for things to pass through and to filter what's passing through and to repel large things from getting through, because you don't want to have those things pass through because it's harder for those to get reabsorbed because the system's not designed for that. So it's not adapted for that sort of thing. So if a cell gets through, the cell can't get reabsorbed. It's just gone. So the system just can't handle it. And so here are some of those structures. Again, you can see what we're looking at here. So the afferent, efferent arteriole, the capillaries. You can see the membrane that's covering it, and that helps serve as like a part of the filtration step to it. Filtering membrane, the membrane itself. And so this is a closer look at it. It looks like a weird candy cane with a moss fungus thing growing on it with a creepy eye. Yeah, this freak. You can see the pores in the capillaries as things pass through, and then all those processes, and those help serve as the filtration membrane and help repel too big of things from passing through and allow small enough things to get inside the cell. Here's another zoomed in version of it, and so you can actually see how thick those food processes are and how they serve as a very strong filtration barrier to prevent large things from passing through. And diving in a little deeper, you can see the same thing here. So you can see the opening, you can see the foot processes, and like the small little spaces that prevent large things from passing along. So they have limitations in the size to funnel the fluid through in such a way that it doesn't allow things to pass through. So this is essentially just like a big like strainer. You like cook pasta and you want to get all the water out of it. You want to keep the pasta in, so you pour it inside of there. And then, or is that a colander? Whatever that structure is. So you pour your pasta in that plastic container. The pasta stays in, the water leaves, boom, there you go. So it's the same thing. It's just modified to allow and prevent big things from passing through. So in this filtration membrane, the large molecules get stuck. They can't pass through. They get taken up by cells. Small things will pass through, forms filtrate, so that will get reabsorbed. And then plasma proteins remain in the blood and help with colloid osmotic pressure, which we've seen about, which helps control the fluid movement in the capillary bed. And so this helps keep you from losing all the water to that space area. So this keeps some water inside the blood and inside those arterioles. You don't want to lose all of it. You want to keep some of it there. It's getting filtered a lot anyway, so it'll eventually get filtered all the way through. So, yeah. Now, the pressures that affect filtration. I'd be remiss if we didn't go through and talk about these again. Who doesn't love a good capillary bed pressure equation, right? So let's do it. Luckily, these are pretty similar. We've learned before, just now in different locations. Where it was HPC, now it's HPGC, where the GC stands for glomerular capillaries. And so, let's see. So this is the hydrostatic pressure in those capillaries. So it's the pressure the blood is exerting on the wall of the capillary bed. So it's the main force that's pushing the fluid inside the blood, that water and solutes out of the blood into the nephron. Is a very high pressure compared to most capillary beds. So as you can see, normally it was a lot smaller than 55, and now this joker is high. And this high pressure exerts just blasting that fluid out of there so it can be filtered. And then the normal capillary bed pressure, if you remember, it was 26. This thing, 55. So, oh boy. And again, we've already covered why that is high. The previous thing on the back was a super soaker. I had one of those as a kid. They were pretty awesome. You got these big tank water guns, and they could deserve some high pressure. I remember I accidentally shot my cousin in the eye with it. He didn't go blind, but it hurt. So I can only imagine that must have hurt. So yeah, I remember like people getting in trouble with pools and stuff. Like, all right, put the gun up. The super soaker. I remember we had like some cool water gun fights. I remember bringing that joker along, and I had some success with that for sure. Anyway, inward pressures. So the things that are preventing fluid formation from occurring. So the hydrostatic pressure that's pushing fluid out, what's keeping fluid in? One of those pressures is the hydrostatic pressure in the capillary space. And so that's the pressure inside the capsule area itself pushing fluid back into the capillaries. This is pretty low. It exists, but it's pretty low. The other pressure is the colloid osmotic pressure in the capillaries. So this is the one that's pulling fluid from the filtrate back into the capillaries, right? So we can see how high that pressure is. So collectively these are 45, but that's less than the 55 of the hydrostatic pressure, which means a net movement of water across the membrane. So there we go. So these are similar terms, but a little different. And so the overall net filtration pressure is the sum of all those things together. And bam, 55 minus 45 equals 10. That means that's your pressure of driving fluid out. So filtration is happening, fluid's going out overall, and that's it. And this is one of the main factors that contributes to your GFR rate. So or GFR, which is your glomerular filtration rate, which is how active and how much blood your kidneys are filtering. This can help determine health of kidneys. And so here we go. So you can see this. So this sums up the pressures pretty well. I think this is pretty easy to compare because we don't have the colloid osmotic pressure of the capillary space, capsular space. So we just have three pressures here. So it's pretty easy, I think. We've already learned the hard way. So this is the easy way. So GFR. This is an important measurement. And so this is the volume of filtrate that is formed per minute by both kidneys. And so this is, evaluated, measured to see health of kidneys. And so it's directly proportional to a few things. One of these is net filtration pressure. So it's just the main pressure that affects the GFR is that the hydrostatic pressure inside the glomerulus. So if that's too high, too low, that can cause, changes there. So if the incoming arterial blood pressure is high, low, that affects the overall net pressure the most. The nephron, the capillary space isn't that important. It's really just the arterioles. Another thing that can play a role is the total surface area available. So the cells can control this by contracting, and that can help change the amount of space available, which will slow down the GFR. And then you also have the filtrating membrane permeability. And so in general, this means that it's much more permeable, so it allows more things to pass through. So as long as that membrane stays that way, then it means fluid can pass through easier and keep up that glomerular filtration rate. Because that's just a reflection of how much water is being pushed out and made in kidneys. So, yeah. So, all right. Next up, how do you regulate this thing? Well, the constant GFR allows you to always be making filtrate and maintain homeostasis in the body at various areas. But the goal of local controls around the kidneys is to maintain the global glomerular filtration rate in the kidneys. So this is important because the GFR affects your overall blood pressure. And so if you have an increased GFR, you're going to have increased your urine output, which is going to lower blood pressure. And this makes sense because if you have a higher blood pressure in general, that's going to increase the pressure inside the glomerulus, which is going to increase the filtrate formation, which means an increased GFR, right? So, I mean, if you link these things together, it makes sense. And if you have a decreased GFR, it means you're going to decrease urine output because you have a lower pressure. Right, so again, it makes sense. Extrinsic controls that regulate this, their main job is to maintain systemic blood pressure, and so the nervous system and endocrine systems can play a role with this. So they're the two main factors that help control the kidneys and the GFR rate. All right, so with the intrinsic controls, this pretty much works and maintains a constant GFR as long as your blood pressure is within this sizable range. If you go out of that range, autoregulation of the kidneys won't work anymore, so it gets a little trickier. So that's kind of rough. There are a couple types of renal autoregulation. Not getting into that really, but they just both deal with blood flow going to the glomerulus. So that's all this is really about. So how much blood is getting to the glomerulus that can be filtered out and form filtrate, which will become pee. The extrinsic controls, the neural and hormonal side of things, we're going to look at those in a little more detail soon. But the purpose of these is to regulate the glomerular filtration rate and maintain blood pressure. Again, extrinsic controls can override the intrinsic controls if you need to increase your blood volume. So maybe times of crisis, you increase blood volume, maybe therefore blood pressure. The sympathetic nervous system is one of the main regulators. And so under normal conditions, the renal blood vessels are dilated and the autoregulation mechanisms are the dominant force. But that's only under normal conditions. When it's not normal conditions, maybe times of stress, then the sympathetic nervous system is activated, or is firing away, and it's going to cause, maybe constricting these blood vessels or whatever needs to be done. So yeah, abnormal conditions. So what we got? So norepinephrine comes in, adrenaline, right? It comes in. It's going to be released by the adrenal medulla, which is right next to the kidney. And it's going to cause vasoconstriction to increase blood pressure. And it's also going to decrease the size of the arterioles that are feeding the glomerulus. So what this does is, by decreasing the amount of blood the glomerulus is receiving, it's going to decrease the GFR. And so, by extension, you're going to increase blood volume and blood pressure and decrease urine formation. This thing here, this is the coolest animal, and one of the coolest animals in the entire world. It's the kangaroo rat. The kangaroo rat is amazing, and then it has super long nephrons in its system, so it does not drink water ever. Instead, it only gets its water from its environment, from the food it eats, and it has such a unique and adapted kidneys system, urinary system for this lifestyle. They form a super concentrated urine, and so that's how they're able to survive and do insanely well with their job. And so we're going to look at the physiology in the part 2 a little more of how that works, so you can understand how this joker is able to pull that off. But man, that thing is so cool. There should be a companion video this week, so I highly recommend checking that thing out. I love this thing. It's such an amazing animal. So it's so cute too. It's a rat, not a kangaroo, but they just call it the kangaroo rat because it looks silly. But then to double touch on something we mentioned before, the Renin-Angiotensin-Aldosterone Mechanism. This is the main one. If you want to increase blood pressure, renin, it leads to an increase in blood pressure. Overall, so there's all those steps, there's a lot of different pathways that are involved. But in short, the sympathetic nervous system activates these granular cells, which then affects the constriction of the arterioles, because these granular cells are specialized to smooth muscle cells that are found in the walls of those afferent arterioles. And so by activating those, it constricts those smooth muscle cells and can control amount of blood flow going to the glomerulus, which affects your overall blood pressure inside the body. And so the other thing that can lead to renin release is some cells called maculodensa cells if sodium chloride levels are low, and that'll increase and prevent filtration formation as well. And so both of those things lead to an increase in blood pressure by factoring in more absorption and preventing filtrate from being lost. And here's a table that kind of sums up all of that. So this is a cool little figure that shows some of those different mechanisms in play, how renin works. We've talked about renin before, so you can kind of see it again. You don't have to explain this too much, just understand that renin plays a role. And we'll probably talk about this, I can't remember for sure off the top of my head right now, but we'll talk definitely a lot more about the physiology of how this kidney functions coming up soon. So yeah. All right, urinary part 1 in the bank, urinary part 2 coming up. Stay tuned.